Welcome back to my channel, and today in this video we are going to study about coupling and repulsion. In crosses for linked genes, the arrangement of alleles on the homologous chromosomes is critically important in determining the outcome of the cross. What it is saying? It is saying that to determine the outcome, that means the phenotype, when we are crossing or when we test cross or when we perform test cross, what will be the phenotype of the cross? That depends upon the arrangement of alleles on homologous chromosome. For example, this is one homologous chromosome. Suppose one gene is present here and another gene is present here. This is gene A, this is gene B. In the homologous chromosome here, it can be again capital A, capital B, or it can be small a also and small b also. So what is the arrangement of the alleles on the homologous chromosome? It will only decide the outcome of the cross. If in this figure only, if we talk about mutants, suppose in a chromosome A is mutant and B is also mutant in this one chromatid. And in the second chromatid of the homologous chromosome, both are normal. But it can also be in the form that in the first chromatid A is mutant, whereas in the second chromatid B is mutant. So it can be like this also. So what is the arrangement of alleles? How the alleles are arranged on the homologous chromosome, it will only decide what will be the phenotype of any cross. For example, inheritance of two linked genes in Australian blowfly, that is Lucilla cuprina. Now, in this example, we are talking about one locus which determines the color of the thorax, purple thorax and the green thorax. Purple thorax denoted by small p, it is recessive character, and green thorax represented by p plus is normal. Similarly, in the second locus, we are seeing the color of the puparium. First one is the color of the thorax, second locus is for the color of the puparium, where black puparium denoted by small b is recessive to the brown puparium, which is denoted by B plus. Clear? So now we know P plus is dominant and here B plus is dominant. So these locus are linked and these are present on the second chromosomes. That means these two genes are tightly packed or these are linked genes. Now suppose a heterozygote parent is crossed with a homozygous recessive parent. What will be the gametes? In case of homozygous recessive, only one kind of gamete will be formed, small p and small b. But in case of heterozygote, we can get two types of gametes, small b, small p plus and b plus. Why we have not got pb plus and p plus b? Why we didn't get this combination of gametes? Because the genes are linked. P is linked with B, P plus is linked with B plus. So we won't get any heterozygote over here. Now in the F1 generation, when we will cross, so this PB with this PB, we will get PPBB. Similarly, P plus B plus with PB, we will get PP plus BB plus. Now again, when we will cross F1 progenies, so we have another PP plus B plus. So what we will get? PB, P plus B plus, or we can get PB plus and P plus B. What we said? P is linked with P and P plus is linked with B plus. So here PB and P plus B plus. But it is not necessary that the mutants will be on the single chromatid of the homologous chromosome. Like I explained in the last slide, if we have two genes A and B, it can be a case that both the mutants are present on same chromatids, or one chromatid will have one mutant, another chromatid will have another mutant. It can be like this. So that is only represented here. PB, P plus, B plus. Or it can be PB plus, P plus B. So when here both the normal, here both the mutant on the same chromatids of homologous chromosome, and both the normal on the same chromatid of homologous chromosomes. These two homologous chromosomes, P and B on the same side, P plus B plus on the same side. So this is known as coupling or cis configuration. When one chromatid contains one mutant and one normal, another chromatid also contains one mutant and one normal, we call it as a repulsion or trans configuration. So cis means, the word cis means same side. Cis means towards same side, same direction, whatever. Trans means opposite. So when both the normal and mutants are on the same side, we call it as a cis or coupling configuration. When they are on the opposite side, we call it as a trans or repulsion configuration. So this is cis, one chromosome has mutant, other has wild type alleles of the gene. In trans, both the chromosome have one mutant and one wild type alleles. Now, this coupling and repulsion only determines the phenotype of any test cross. Now, let's see the alleles in coupling configuration. Suppose PB, P plus B plus is crossed with recessive partner. Now, from here, we'll get only one type of gamete. But from here, we will get four types of gamete. Because they are linked genes, so the number of non-recombinant will be more. But some recombination is always allowed. So we can get recombinant gametes because the parental is PB, P plus, B plus. So the recombinant will be P, B plus, P plus, B. This one. So we will get recombinants as well as non-recombinants gamete. But the number of non-recombinants will be more. This we already studied in the last video. Now in the F1 progenies, what we will get? As you can see, this gamete combined with this gamete will get PB. This gamete again combined with this gamete will get PB, P plus B plus. This gamete combined again with this gamete will get PP, PB plus. And this gamete combined again with this gamete will get PB, P plus B. So this is parental type. This one you can see here. This is also parental type. You can see here the parent. So these are non-recombinants and more in number. Rest recombinants less in number. Now when we see the cross in case of repulsion configuration. Now in the repulsion configuration, as you can see, the arrangement of alleles are different. In coupling it is PB, P plus, B plus. In repulsion it is P, B plus, P plus, B. So in case of coupling, this is parent. In case of repulsion, this arrangement is parent arrangement. So we will get more of non-recombinant gametes. What is non-recombinant gamete here? PB plus and P plus B. This combination is recombinant in case of coupling configuration. But this combination is non-recombinant in case of repulsion. Because in repulsion, we have one wild type and one mutant allele on one chromatids of homologous chromosome. Similarly, we'll have recombinants as well. So here, if you see the recombinant, it is same like non-recombinant in case of coupling. Where is non-recombinant? Here, right? So, which is non-recombinant in case of coupling is recombinant in case of repulsion, which is recombinant in case of coupling is non-recombinant in case of repulsion. So we have this now for F1 progenies. Again, combine these two, then these two, then these two, then these two. Here also we will get more of non-recombinants and less of recombinants. But if we see the arrangement, the non-recombinants here are similar to recombinants in case of coupling. So always remember the arrangement of alleles will determine the outcome. The arrangement is in coupling configuration or in repulsion configuration. Both are completely opposite of each other. What we will expect in coupling configuration will get different result in case of repulsion. What we will see in repulsion, exactly opposite will get in coupling configuration. So whether the alleles are arranged in coupling or repulsion configuration will determine the outcome. So as the genes are linked, recombinant will be less compared to non-recombinant progenies in F1 generation. This we already studied in the last videos.